What? 30 right now.
Let's give it 1 minute.
No.
Well.
If I see a typo on each slide, don't say anything. Whoops. You can raise your hand. Oh, is this being recorded? Yeah. Okay, is that okay?
Okay, welcome everyone. Thank you to everyone for coming. And thank you to Maggie's parents, her family, her partner, Michael, her sister, Emily. Maggie has an identical twin, so be careful who you congratulate afterwards. And Emily's boyfriend. And thank you to all of you for coming. I also really want to thank Maggie's extended network of friends and family for all the support over the years and getting to this point. So Maggie came to us from Kalamazoo College, which she enjoyed greatly, and she did research there on microbial ecology, which she liked a lot, and she got the research bug. So she decided to come to Wayne State, and I had the great good fortune to have her rotate in my lab and then join my lab. So in characterizing her, I would say she's very collaborative and she's a really good lab member. She's helped and trained people in my lab, and she's trained a lot of people throughout the department. Everybody that uses the qPCR machine comes to her, and including me. So she's now training me on how to use the qPCR machine and how to do luciferase assays. So she's been curious, she's collaborative, she's curious, and she's been a joy to have in the lab. So she selected a project that deals with something that we've long known and suspected played a role in dosage compensation, and that's the architecture of the nucleus and the structure of chromatin on the X chromosome. And she initiated a series of studies looking at candidate genes to try to figure out if any of these genes actually participated in this process. And I have to sort of characterize her work as maybe a little more than we bargained for. It turned out that we found a lot of genes that participate in this process, but in a couple of instances, they did remarkably surprising things. And so I will let Maggie tell you about it.
Okay, thank you for that introduction, and thank you all for coming to my talk today. I'm using that. So I'll be telling you about my work looking at factors that are involved in X identification, a process that is required for male survival in Drosophila. Okay, welcome to the Drosophila karyotype. Drosophila have 4 pairs of chromosomes, 3 autosomes, and 1 pair of sex chromosomes. Drosophila are just like humans in that the females have 2 X chromosomes and the males have 1 X chromosome and 1 Y chromosome. But this creates an imbalance of expression of genes off this X chromosome between these 2 sexes, and this needs to be compensated for. In mammals, females will compensate for this difference by inactivating one of their 2 X chromosomes completely to match that of the single male X chromosome expression. This is done in part by this long non-coding RNA called exist that coats the inactive X chromosome and recruits factors to silence this chromosome. In C. elegans, the organism that has 2 X chromosomes is a hermaphrodite. These hermaphrodites will reduce the expression of both of their X chromosome by approximately 1/2 to equal that of the 1 male X chromosome. However, in Drosophila, the male will perform dosage compensation by increasing the expression of its single X chromosome by approximately 2-fold to match that of the female X chromosomes. This is done in part by these long non-coding RNAs that coat the X chromosome to cause this increase. They're called ROX1 and ROX2, and they are redundant in function. There is a feature shared amongst many different species of dosage-compensated chromosomes, and that is their localization and structure within the nucleus. In C. elegans, the hermaphrodites that are dosage compensating both of their X chromosomes, you often find them sort of compacted and localized near the periphery of the nucleus. And then you can see upon knockdown of a certain factor required for C. elegans dosage compensation, you see a loss of the structure and localization within the nucleus. In mammals, here stained for exist, which is coding that inactive X, mammals often find their inactive X chromosome, the females, near the nuclear periphery as well. Similarly, in Drosophila, here I have staining for MSL, which is the complex important for dosage compensation in Drosophila. You can see it binding to an X chromosome territory that appears to be near the periphery by the nuclear envelope. Upon RNAi of a nuclear pore protein, you see loss of the staining of the MSL complex to this X territory, and no longer have that localization of that X chromosome and staining at the nuclear periphery. So let me tell you a little bit more about this MSL complex. Like I mentioned, this complex is made up of 1 of 2 ROX RNAs. These are important for localization of this complex to the X chromosome. Other important features, this MSL3 protein, which binds into H3K36 trimethylation marks to spread this into actively transcribed genes. MOF is males absent on the first. This is a chromatin modifier. It places acetylation marks on the chromatin so that it can open up the chromatin at these active genes, allowing for an increase of expression. After assembly of this complex can localize to this adapter protein called clamp, which is bound at the DNA to localize this complex back to the chromosome. We know quite a bit about how this mechanism is working. The ROX RNAs are transcribed off of the X chromosome. Once it's transcribed, the rest of the MSL complex can assemble, and upon assembly can be recruited back to that clamp protein, which is bound at the X chromosome at these chromatin entry sites, or CESs, as I will call them for the rest of the presentation. These chromatin entry sites contain GA-rich sequences called MREs, or MSL recognition elements, to which clamp is binding. So now that the MSL complex is localized back to the X chromosome, this complex can spread using the MSL3 binding properties co-transcriptionally into the body of active genes, where it will then place acetylation marks into this gene, causing an increase of expression. So although this is a very nice model of how this is working, there still remains one big question that remains unexplained, which is the main focus of study in the Meller lab. And that is that these MREs to which clamp is binding are present both on the X chromosome and on the autosome. Not only that, but clamp is able to bind to MREs at the autosome as well. Yet somehow, in males where the MSL complex is expressed, the MSL complex is only localizing to these clamp proteins at the X chromosome. So the complete picture of how it's able to identify and localize to this X chromosome remains unexplained. There are several hints that have been gathered over the years, though, of how this might be working. The first is, as I mentioned, that ROX RNA that is important for proper localization of this complex to the X chromosome. Here I'm showing you polytene chromosomes from male salivary gland squashes. In each of these cases, ROX2 has been deleted, but ROX1 in this case is wild type, and since they are redundant in function, when we stain for MSL1, you see very faithful and complete staining of the X chromosome, or of the MSL complex to the X chromosome, resulting in 100% survival of these males. With ROX2 again deleted, but a mutation in ROX1, this results in a reduction of staining of the MSL complex to the X chromosome. You also see some staining to autosomal sites as well, some mislocalization of that MSL complex, and this results in a reduction of survival of these males to about 45% in this specific mutant. And under an even more severe mutation, you don't see any sort of coding of an X chromosome whatsoever, a lot of mislocalization to autosomes, resulting in an abysmal amount of male survival to about 5%. Interesting about these ROX genes, they are both encoded on the X chromosome, ROX1 and ROX2. They also both contain internal CESs within their sequence. If you move ROX1 to an autosome, we find MSL complex is able to localize to ROX1 at that autosome, as well as spread into nearby active genes and allow for the upregulation of expression of these genes. However, if you remove ROX1 and ROX2 from the X chromosome and retain this ROX1 on the autosome, you do still see the MSL complex binding to this autosomal ROX1 at the CES, spreading into nearby active genes, but also the MSL complex is able to travel back to the X chromosome and upregulate genes here, resulting in male rescue. So this indicates that the chromatin entry sites and the ROX gene itself is not alone marking the X chromosome for localization, because without the ROX genes here, the MSL complex is still able to come localized to the X. So we still don't have a full picture of how this is able to work. There is another cis element that our lab is interested in studying, and that is these 1.688 satellite repeats that we call the 1.688X. These are satellite repeats that are AT-rich, about 359 base pairs in length. They're in the euchromatic end of the X chromosome, and they're stained here in this darker color. Here I'm pointing out the 1.688 repeat at the 3F cytological position. If you move this repeat to an autosome, similar to moving ROX1 to an autosome, we find MSL recruitment to this repeat on the autosome, as well as an increase of expression of these genes nearby. Another known feature of these repeats is that they are transcribing RNAs from both strands of these repeats. Former student Debashish Renan was curious about this. Double-stranded RNAs could turn into siRNAs and be used through the siRNA pathway. So he wanted to explore how these repeats might be involved with the siRNA pathway. He found that upon this particular mutation, this excision 33 particular mutation, there's normally about only 20% male survival. But when he induced more expression of 1.688X siRNA, he saw a significant increase of survival of these males, indicating that the siRNA pathway may be acting through these repeats to aid in this X recognition. Debashish also wanted to further explore this and solidify his thought that the siRNA pathway is involved with this process. To do this, he wanted to look at siRNA pathway mutations along with the ROX gene to see if they are genetically interacting. So genetic interaction is identifying genes that they aren't physically interacting, but they are maybe acting in parallel pathways or acting in complex with each other. He utilized this concept of synthetic lethality to study this. So to briefly describe this synthetic lethality, if you have two genes that you are observing, and they are both at wild type levels of expression, you would obviously expect this organism to live. If you reduce the levels of expression of one gene and that organism lives, or reduce wild type levels of expression of the other gene and that organism lives, but reduction of both results in a sick or dead organism, this indicates that these two factors, these two genes that we were looking at, are genetically interacting. Debashish used this with mutations in ROX1 and ROX2, along with different mutations of proteins involved in small RNA pathways. Debashish found with mutation of proteins in the siRNA pathway, there was a significant reduction of male survival, indicating that there was a genetic interaction between several of these siRNA proteins and the ROX genes. You might think this is curious because, as most of us know about, the siRNA pathway is a generally silencing pathway. It's reducing the expression of genes, but dosage compensation is a large upregulation of genes on an entire chromosome. So how might this generally silencing pathway be aiding in dosage compensation? Former student Nikita Deshpande was also curious about this, and her studies led her to this hypothesis. So we have the RNAs being transcribed from both strands of these repeats. This can create base-paired double-stranded RNA. This double-stranded RNA can be processed by these siRNA pathways that I had just shown you had a genetic interaction with ROX1 and ROX2, and they're produced into small interfering RNA or siRNA. These siRNA are loaded onto the AGO complex and guide this complex back to the actively transcribed repeats and can recruit other factors such as SUVAR39 back to these repeats as well. SUVAR39 places H3K9 dimethylation marks. And Nikita did indeed find an enrichment of these H3K9 dimethylation marks around these repeats. And not only that, but further induction or expression of these siRNA constructs from the repeats increased this enrichment of these marks around the X. So her theory is that perhaps the heterochromatic state that these are creating, or just the marks themselves, are sort of acting as a flag or a beacon for the MSL complex to be guided back to this X chromosome. And that's how this negatively, generally thought of as a process for negative expression levels, could be increasing expression through dosage compensation. So all this evidence we've gathered has led us to our lab model. Our model is that there are two different cis-acting pathways collaborating for dosage compensation. The first is those CES through which the MSL complex is operating. The other is the 1.688X repeats through which the siRNA pathway and chromatin modifiers are somehow acting to cause X recognition, which will allow for the MSL complex to upregulate genes on the X. Still does not paint the full picture of how this is operating, right? So to fill the gap, I was interested in, for my study, is to hark back on what I'd mentioned earlier about proteins that are, or chromosomes that are dosage compensated, and their localization within the nucleus and their structure within the nucleus. So my hypothesis are that factors perhaps bound to repeats might help mark the X, but also different factors that are aiding in chromatin organization within the nucleus or subnuclear localization are aiding in this X recognition. So to explore my hypothesis, I first came up with a long list of candidate proteins that I wanted to explore to determine if they are playing a role in dosage compensation. I'll tell you briefly about these proteins now. Here we are inside the nucleus. First protein is SAF-A, which is a scaffold attachment factor A. It binds along with RNA and it binds to chromatin, and it creates this chromatin mesh that maintains an open chromatin structure. Imitation switch is part of a nucleosome remodeling complex that can slide nucleosomes around. It's also been, this complex that it's a part of, the NERF complex, has also been previously implicated to be interacting with the chromatin entry sites. So we think this will be a strong candidate to be participating in this pathway. D1, which is a AT hook protein, so it has these protein domains called AT hooks bind into AT-rich repeats and AT-rich regions. It is known to bind into the heterochromatic end of the X chromosome, which has 359 base pair repeats similar in length and sequence to the 1.688X repeats on the euchromatic euchromatic end of the X chromosome that we are interested in. So we thought this would be a promising candidate to add to our list. Also, it's important for maintaining a chromocenter, which holds together a lot of heterochromatin within the nucleus. So important for structure within the nucleus as well. HB2 is a heterochromatin protein, also AT hooks binding into satellite regions. There seems to be some mysterious link between heterochromatin and dosage compensation that we've been investigating in the lab recently that we don't know how to explain. So HB2, more heterochromatins added to the list. See if they're participating. Modifier of MDG4 and CP190. These are both insulator proteins. They're bound at several different types of boundaries, and they also are thought to maintain long-range interactions of chromatin. NUP153, which I had previously shared with you, loss of NUP153 caused a loss of localization of the MSL complex to the X chromosome. Nuclear pore proteins are also known to make contact with chromatins, makes it a strong candidate for use in our study. And lastly, lamin B, which is a nuclear lamina protein, the nuclear lamina at the periphery of the nucleus, also known to make contact with chromatin as well. So with my list of candidates, I had a gamut of different tests I wanted to run them through to determine if they're operating in X recognition. First of which, male preferential lethality. Since males are the only ones doing dosage compensation, if mutation or loss of one of these factors results in a significant phenotype in specifically males, then this might indicate that it's operating in this male-only process. Next, a genetic interaction with ROX1 and ROX2, so similar to what Debashish I had shown you done to find the siRNA pathway proteins, seeing if my proteins of interest are also genetically interacting with the ROX genes. And lastly, using a functional assay that former student Maggie had developed to determine which recruiting pathway each of these factors might be operating through. A very useful tool for the trade are these RNAi knockdown fly lines. These are lines you can order online and they're readily available. They're available for virtually any gene you might want to test. Sometimes there's multiple per gene that you want to test, which is good because the level of knockdown of these can be variable. So the first data that I will show you is using these RNAi knockdown lines to determine if there's any specific effect of knockdown on male survival. So comparing the levels of knockdown males to control for all my different proteins of interest, I've sorted them into the categories here. So heterochromatin proteins binding with AT hooks, other factors involved with chromatin binding and remodeling, proteins that are found at the nuclear pore or nuclear envelope, and then those insulator proteins as well. Upon comparison of male and female survival, I did not see any significant difference in survival between the sexes. There were some cases like SAF-A and CP190 where I did see a reduction of survival of both sexes, but there was no significant difference between the two. So although I was not able to find a male-specific effective knockdown, I can still use these lines with that simultaneous ROX mutation for to determine if there's a genetic interaction with the ROX genes and my factors of interest here. So upon simultaneous mutation with ROX1, upon knockdown, I found several different lines to have a significant reduction in survival of males. These ones in particular. So I have some that are AT hook proteins for heterochromatin proteins, proteins involved with the structure of chromatin or the state of chromatin, and a nuclear pore protein. So interesting to have a diverse range of proteins with diverse functions showing up to be participating in dosage compensation somehow. But I want to know more. I want to know how these different proteins with their very different functions through which pathway they might be operating. As I mentioned, Maggie developed an assay to discover just this. Moved a candidate recruiting element or several candidate recruiting elements from the X chromosome, since the X chromosome is already littered with recruiting elements and recruitment of the MSL complex, moving them to an autosome along with a reporter gene. She used firefly luciferase. And we can compare this, or we can compare MSL recruitment to this region and upregulation of this reporter gene to a normalizer that does not have a recruiting element. So these are the 4 constructs that Ream designed. First of which is just F-Luke, that firefly luciferase alone, which will create a baseline level of luminescence. With the addition of a recruiting element like ROX1 that has that internal CES, would recruit the MSL complex, cause upregulation of F-Luke, you see an increase of luminescence. Or F-Luke with the 1.688x repeats, also recruiting the MSL complex, increase of luminescence. She also designed a 4th construct that has both ROX1 and the repeats to determine if there's perhaps an even more increase of luminescence. This is Ream's data validating the use of her constructs. She did this in embryos. So for these assays, we compare the levels of F-Luke activation divided by the activity of R-Luke, which is a normalizer gene found on a different autosome. With no recruiting element, we set this as our baseline as 1. And then Ream saw with the addition of ROX1, the CES, a significant increase of that F-Luke expression. And again, with repeats and both ROX1 and the repeats, a significant increase. So this indicated to her and to all of us that wanted to use her assay that this assay can be used to measure recruitment to the autosome and upregulation of a firefly luciferase reporter gene. We can now use this assay along with knockdown of proteins of interest to see if there's any block of activation of this firefly luciferase expression to determine through which pathway these proteins may be operating. Ream also validated her assay first with knockdown of MSL2. MSL2 is important for the assembly of the MSL complex. So ideally, we need this MSL complex for any sort of upregulation of genes. And Ream did indeed find that with MSL2 RNAi, there was a significant block of this activation of the firefly luciferase gene in the presence of the recruiting elements. So this is sensitive to knockdown of important factors to dosage compensation. Ream also validated that the system could be more specific in identifying through which pathway this might be working by knockdown of AGO2. AGO2 is that protein involved with the siRNA pathway important for 1.688x recruitment. Upon knockdown of AGO2, she finds a block of activation only in the presence of these repeats, validating that we can use this system to determine a specific pathway through which these factors are operating. I used Ream's assay on adult flies rather than embryos. Under control conditions with no RNAi, I saw similar levels of increase of firefly luciferase activity. I next wanted to use a control. I used both yellow RNAi and M-cherry RNAi. Yellow is a gene involved with just body color of the fly. M-cherry is not even a Drosophila gene. And this was to determine that the RNAi process itself is not affecting the firefly luciferase activity. I did not see any significant difference with the RNAi knockdowns for the controls, which was what we expected. So first, I'll show you data now for imitation switch. Imitation switch is that protein involved within the nerf complex that remodels chromatin and slides nucleosomes around. Also previously been implicated through the ROX1 and CES pathway. Upon knockdown of imitation switch, this RNAi line had kind of a moderate level of knockdown, about 42% of transcript of imitation switch left over as compared to wild type. With imitation switch knockdown, I see a very staggering and significant decrease of expression of firefly luciferase in the presence of ROX1. This validated that this protein we had predicted to be operating through ROX1 and the CES is indeed doing as we expected. Next, I used this assay to explore the roles of SAF-A and HP2. HP2 is a heterochromatin protein binding with AT hooks into AT-rich heterochromatin. With quite strong knockdown of HP2, I actually saw a significant increase regardless of the recruiting element situation, so in all cases. And alternatively, with SAF-A RNAi, SAF-A is that protein that binds with RNAs as well as chromatin to create a chromatin mesh to create an open chromatin environment. Very strong knockdown of SAF-A caused a significant decrease of expression of firefly luciferase, regardless of the recruiting element present. So this indicated to me that perhaps these factors that are involved with chromatin state are kind of operating in a more general manner, and that male flies that are dosage compensating need this chromatin state to effectively do dosage compensation. So what I've learned so far through Ream's assay, imitation switch is operating through those chromatin entry sites, which is as I had expected. HP2 and SAF-A are altering expression regardless of what recruiting element is present, but I did see them genetically interacting with the ROX genes earlier, so they are playing some sort of a role. And perhaps these males that were deficient in ROX or deficient in dosage compensation because of their ROX mutation are just more sensitive to chromatin disruptions. All right, next is D1. D1 is also that AT hook protein binding into the heterochromatic end of the X chromosome. With very strong knockdown of D1, I found a very significant decrease of firefly luciferase activity with ROX1 and the CES, but no change in activity with just the repeats. This was a bit unexpected because ROX1 is an AT hook protein binding in repeats very similar to the 1.688X. I expected that it would perhaps be operating through this pathway, so it was very surprising to find this significant reduction of firefly luciferase activity with the CES present. I was curious to determine how knockdown of D1 might influence or change MSL binding throughout the genome. I also utilized polytene chromosome staining to determine this. So under wild-type conditions, as shown previously, the MSL complex is able to bind faithfully to the X chromosome. With this particular mutant that I used for this test, which is excision 40, there's a very high survival of these males. They still have very good staining of the MSL complex to the X chromosome. There does seem to be some autosomal sites, but it's kind of no consequence to them because they have nearly complete survival. But it does create a sensitized background. Upon knockdown of D1, I did not see any significant difference in MSL staining to the X chromosome. And with simultaneous D1 knockdown and ROX mutation, the MSL staining also maintained at the X chromosome. Since these can be very variable from organism to organism and nuclei to nuclei, I also quantified these looking at fraction of nuclei for several different categories. Kind of most important to look at is wild type X under wild type conditions and ROX mutated conditions, as well as D1 knockdown and the simultaneous condition here. I see mostly wild type staining of the X chromosome. These other factors here, like binding at the chromocenter, minimal to no staining of the X chromosome, autosomal sites of staining, or the telomere are often features that we see when we find mislocalization of the X chromosome. So with these ROX mutations, we do see a little bit of this mislocalization, but for the most part, still wild type. So overall, what I've learned so far about D1, loss of D1, as I had seen, it is enhancing this ROX1, ROX2 lethality, and it's unexpected that it's acting through the CES. Although there's no sort of gross effect on looking at the polytene chromosomes on MSL staining, there is obviously some sort of effect on MSL localization because with knockdown of D1, we do see that it impairs expression of that firefly luciferase through the CES. So it's operating a little more at the gene level rather than at this gross level. All right, CP190 is that insulator protein that is managing likely long-range interactions of chromatin and binding into boundaries as well. With kind of moderate levels of knockdown of CP190, I actually saw a significant increase of expression with the repeats present no significant change with ROX1 present, indicating that these CP190 is acting through the 1.688X pathway. Not exactly sure why it's increasing, but we do find that interesting. But looking at MSL staining upon CP1 knockdown, so comparing again to the same control conditions as I'd shown in the previous slide, CP190 knockdown alone caused a lot of mislocalization of the MSL complex and lower wild type staining of that MSL complex. So CP190 alone is able to cause an effect, or knockdown of CP190 alone, able to cause an effect on gross MSL staining. And then in this sensitized background with the simultaneous ROX mutation, these males were very sick and unhappy, and it was very hard to get very good preps of these because of that. Their salivary glands were very tiny. You don't see any staining of wild type X, all sort of mislocalization if you can see much of anything at all. So I guess that's validating that genetic interaction between these two. So we were baffled to find this increase of expression through the repeats, although it is interesting to find that is operating, CP190 is operating through the repeats. It is enhancing this male lethality with the ROX mutations, and it's severely altering MSL localization. CP190, we do know, is very promiscuous in that it has a lot of binding partners, a very long list. So perhaps knockdown of CP190 is releasing some of these binding factors that are somehow able to increase this activation of this F-Luke gene. It's a theory. And lastly, NUP153. NUP153 is that nuclear pore protein that had previously shown to have an interaction as RNAi caused a mislocalization of the MSL complex. I had sort of a weaker RNAi knockdown here, but upon RNAi knockdown, see a significant decrease of expression through ROX1 in the CES. Not in any other case, though. Upon NUP153 knockdown, the X does stain wild type with the MSL complex. However, with ROX1, ROX2 simultaneous mutation, I did see a reduction in that wild type S and a lot more mislocalization to areas of the autosomes, validating again this genetic interaction sensitized to this ROX mutation. This was a promising find, as NUP153, as I mentioned, has been previously implicated in dosage compensation and through ROX1. Well, now we know it's operating through ROX1 and the CES, so I can add that to our knowledge of NUP153's role in dosage compensation. So after the exploration of all of my proteins, I'm now able to add them to our lab model. HB2 and SAF-A, although not able to determine operating through a specific pathway, they seem to be important for this function as a whole. Through the chromatin entry sites, I see imitation switch and D1 and NUP153 operating. And then through the X-linked repeat, we have CP190. Okay, so things that I've learned from my experimentation. I learned that this testing of candidate genes and through this process is a very productive approach to find many factors with many different functions and their role in dosage compensation. I found that many different architectural factors are involved in recruitment through both the 1.688X pathway and the CES pathway. Although we don't know exactly how this is working, we predict that some of these influences may be indirect. D1 is binding into a large block of heterochromatin on the X chromosome. Perhaps reduction of D1 binding here is releasing factors or disrupting chromatin in a way that is causing this disruption through ROX1 and the CES. And as I had mentioned, CP190 is also interacting with a lot of factors, so maybe its impact is also indirect, and with some of these factors under many different nuclear processes are also impacting dosage compensation. The chromatin entry sites and the CES rely on different aspects of nuclear architecture. So they both have nuclear architecture proteins, but all these proteins have very vastly different functions. So it's just interesting to see that the complexity of nuclear architecture's effect on dosage compensation. This still leads to a lot of open holes that would be very interesting to fill, I think. First of which is the role of nuclear pore proteins. There are several other nuclear pore proteins. One of them is called Megator. Megator has similarly to NUP153 been found to disrupt MS cell localization upon knockdown. So, and there's other nuclear pore proteins as well to see if they're also influencing dosage compensation, or is there something special about those two? Is the peripheral localization to these nuclear pore proteins important to finding the X chromosome? There also, literature shows that there is some sort of interaction between insulators and nuclear pore, where insulators bound to chromatin can bind to nuclear pore proteins. So is that interaction between these two? Since I do find insulators and nuclear pore proteins important in dosage compensation, is that interaction also important? Also testing CP19D interactors that are mediating very nuclear processes. This is just a few of the sort of functions that CP190 interactors have, involved in insulation, involved with cohesin, members of the mediator complex, so with gene expression, and also facilitating those long-range interactions. So sparsing out what part of CP190 action or its binding partners' actions is important for this dosage compensation. Okay, if you stop paying attention, you can start paying attention again. It's everyone's favorite slide. Okay, first and foremost, I'd like to thank Dr. Meller. Thank you for letting me join the lab. This was a really fun project, and I really learned like what a great world it is inside the nucleus from you. Thanks for teaching me how to be a good scientist and science communicator. If you do any fly work, you know that at the very beginning of the day is the best day to sit down at the fly microscope and collect virgins and do your fly work. So Dr. Miller and I spent many, many hours together over the fly bench in the morning, every morning, chatting about data and science and hypotheses, but also talking about travel and our cats or the Barbie movie or literally anything. So I really treasure that time together, and thank you. Thank you to my committee members who are all here, Dr. Pyle, Dr. Ansari, and Dr. Krawitz. Your input was very valuable. Thank you for helping me keep things on track and keeping me motivated through my project as well. It's nice to have people who you feel like they believe in you. It's very important. My lab members, I'm very thankful to have been put in a lab with these people. Here today is Reem and Marissa, who've become some of my best friends. We just have the best lab environment, and I'm so lucky that I get to have you guys. Thank you for your help with things inside the lab and also just your friendship beyond. Other lab members who have moved on, Sudeshna and Nikita and Milo, who I still talk to all the time, and thanks for their help in the lab as well. Our lab meeting members, a very fun lab meeting group with the Pyle and the Ansari lab, and getting to know all of you in the lab. In those labs. Thanks for listening to me at lab meetings, and it was really interesting to hear what you guys had to say as well. All the biology professors and faculty that I encountered over the years, the people who taught me, people who I taught under, encounters at maybe the retreat, thank you for your input and your motivation as well. The staff as well, the grad students always have questions, the staff always has the answers, so thanks to all the formidable women in the office. The Van Berkom lab, when at the crunch time, when all my options for a fluorescence microscope were damaged by fire and water, the Van Berkom lab came in clutch with a microscopes I could use, so thank you to the Dr. Van Berkom in the lab. Our funding resources, NIH grant that Dr. Meller had earned. I had earned a summer dissertation award as well, and also teaching assistantship. Getting to teach was a really great pleasure and joy. It gave me a skill and happiness that I didn't know I could find through teaching. The Bloomington Drosophila Stock Center for all keeping and maintenance of these beautiful fly stocks, and families and friends, some of which I put here. I'll get to this. Thanks to all the SciPol ladies, particularly Emma and Brenna and Katie. Being in part of SciPol was one of the best parts of my experience here, and I'm glad I get to have you guys as friends as I move forward too. And the work that we did just really lit a fire inside of me that I really needed at that time, so thank you. These are my cats, Calliope and Nereus. They're the best cats, and if you have cats, you just gotta let me have it today. These are the best cats. This is Nereus helping me with my dissertation the night that it was due. He was helping me use my mouse. So perhaps not the most productive to have around, but they help in spirit. I'm so thankful to have lived in the Detroit area so I could have friends from before and friends that I've made now. They've been an absolute joy to be around and really lifted my spirits in times that I needed them. So thank you. Also, I have my family here, my mom and dad, Kelly and Steven, my sister, yeah, she's my twin, Emily, her boyfriend, Richard, and my partner, Michael. Could not have been standing here without all of you today. Thank you for everything, and I'd be happy to take any questions.
Yeah, wonderful, very instructive. And I have many questions, but I would like to focus on CP190.
Okay.
Each video posting the program correctly, right? So what is known of how abnormal the chromosomes look like in vivo in the presence and absence of protein?
Of CP190?
Yeah.
Yeah, I mean,
So then related to that, so my understanding of these beautiful chromosomal squashes is they're moved from 3D to 2D.
correct.
So would there be a way to look at how these chromosomes look like in the absence and the relation?
Right, yes.
across?
Yes, I know lack of CP190 since it's maintaining these long-range interactions. I guess it's been found that obviously without CP190, some of these long-range interactions, but I don't know much beyond like what it looks like inside the nucleus without it, besides just the interaction, loss of some of these long-range interactions. You're right that a polytene chromosome squash, returning it into 2D, would not be a way to explore this. You would have to kind of probably do a whole nuclei approach rather than squashing these chromosomes. Also, maybe not in polytenes as well. Maybe because you have that higher levels of replication, maybe you'd want a more conventional cell rather than a polytene nucleus.
Does that technology exist? So that they can apply?
I mean, I've seen really cool stuff done with like exploration of X chromosome territories using like painting of chromosomes and stuff like that. So perhaps that could be of interest, but yeah, it's a good thought. Yeah.
So I gotta commend you on your super channel.
Yeah.
I had a question, I'm sure Dr. Powell asked this. With you, but
Yeah.
X specific genetic modifications.
X-specific like modifications, yeah. I mean, as I mentioned, Nikita does see those H3K9 dimethylation marks enriched around these repeats, so that is X-specific. It's mostly that, I would say, but yeah. So that in the presence, I mean, it's different because the X chromosome is different in structure, and that it like where's the heterochromatin, where's the euchromatin and stuff like that. So that would be more specific of like marks in the euchromatin versus the heterochromatin. It's kind of split in half that way too, so that might be different than the autosomes as well.
Or maybe it's make the autosome.
Autosome modifications? Yeah.
X specific.
Yeah. Perhaps, yeah. No, that's a good thought. I mean, we talk mostly about those H3K9 dimethylation marks around the repeats, which are special and enriched in that region. So that's what first comes to mind for that. Yeah. Yeah.
Yeah, it was a great talk. I just had a question about your NUP 153. When you have like the ROX1 CES and you have the NUP 153 knockdown, you see that like market reduction, right?
Yes.
But you don't see that when you have the ROX CES and
the repeats.
yeah. Is there a specific reason for when they're both there, you don't see that reduction?
My only thought is perhaps this impact of NUP 153 loss on the repeats is not necessarily strong enough to overact recruitment through the repeats that you see with that combined one, right? We don't really have an answer for that. We haven't explored that, but that's my thought. Yeah.
wonderful.
Thanks.
And I always think like genetics are for very smart people. So I don't think genetics. So I want to ask a very naive question. So here you use most of like archive to order and all this see the. So I was wondering if it's possible to do those. Can you change always? And once you were you going to.
Oh, well, you don't want too much overexpression, right? It maintains, because as you can see with the use of these constructs, when you move it to the autosome, sometimes it's causing like 4, 5, 6 times expression rather than just that double of expression we expect with the MSL complex. So I think there's systems in place on the X chromosome of males to keep it at 2X and not go up. When we move it to the autosome, that's probably not there, these fail safes to prevent it from above 2 times expression, right? So I don't know if we'd want a super male like that. It would probably not be great. Interesting thought. Yeah, overexpression of genes. I don't know, maybe it would have like the opposite effect. It's not something I've considered, but since it's on an autosome and it won't affect this male's important X chromosome thing, then maybe it would cause like kind of an opposite. Thanks for the question.
Any more questions? Are there?
Sure. So at the beginning, you explained to us that our system is different from that of the worms.
Sure.
That we study.
Mhm.
So how old is the insight of whether that's true for all flies or all insects?
Drosophilids, yes. That's a good question. I don't know, not evolutionary biologist. I don't know much beyond Drosophilids, but definitely in Drosophilids, it goes that far. Do you know how beyond? Yeah.
Hey,
It's
you're
me.
the candidate.
I thought it's an interesting question. I'd like to know the answer too.
But there's rumor that mosquitoes have different choice of genes for X chromosome
Mm,
compensation.
Yeah.
Yeah.
apparently.
The sex chromosomes themselves are quite rapidly,
Mm,
hence the systems.
mhm.
Are there any questions?
Thanks.
Okay. Any more questions? Okay.
Thank you.
everyone for
Thanks.
coming. And we will have a reception in the museum in about an hour.
No. Thank you. Thank you. It felt pretty good. Seeing who's. Okay, good. Okay, now the grilling.
Want to take a break before the Inquisition?
I could use this. I could use a sip of water. What do I need to do?
Stop the
Oh,
recording.
I'm so sorry.
Then you can probably end this whole meeting.
Mm.
Do you want me to bring anything upstairs for you?
Oh, I think I'm good. I'll just bring it up when I'm done. Okay. Thank you.
You like crushed it.
Thank you. Okay, my